Well, thanks everyone for being here. I'm
Okay.
gonna go, yeah. My name is Lindsay Olson, and I am the Farmer Training Program Manager at GreenWave. I live in Homer on Supiaq and Denina land. And GreenWave, I think many of you are familiar with, but for those who haven't interacted with us before, we are a nonprofit based in New Haven, Connecticut. But we have staff and board members around the country and in Alaska as well. And we are dedicated to training and supporting regenerative ocean farmers in the era of climate change. And we offer a mix of virtual and hands-on trainings like this one, as well as one-on-one support. And then we recently launched an online platform to help seaweed farmers establish businesses. So I'm really pleased to welcome you all to today's workshop on kelp Sorus ID and collection, specifically for kelp farmers in Prince William Sound. And today's workshop is being hosted in partnership with the Native Conservancy and the Chugach Regional Resources Commission. And we're going to go over the basics of species identification and then best practices for Sorus collection and handling of the 3 commonly farmed species in Alaska. So sugar kelp, Saccharina latissima, ribbon kelp, Alaria marginata, and bull kelp, Nereocystis leuciana. Before I jump into the content, I'm just going to go over a couple Zoom logistics. So today's webinar is being recorded, but it is being recorded in speaker view. So you will only be recorded if you ask a question and those who are speaking. So we do ask, I notice a lot of you kept your cameras on when you joined. It's really nice to see who's in the room. So if you have a stable Wi-Fi connection, we ask you to keep your camera on. But if you are having trouble hearing us or seeing the slides, try turning your camera off to improve that connection. And we also really want this workshop to be informal. So really the whole point of today's convening is for everyone to get their questions answered around Sorus ID and collection. So if a question comes up, please ask it. You can do that in a couple ways. You can type your question into the chat using the chat function at the bottom of your Zoom screen. And I will pause Tamsen to ask it of her or hold it for a break in the presentation and then ask questions in the order they came in. And you can also use the raise hand function. You do that by going to the bottom right of your screen and clicking on the reactions button and then click raise hand. And that will let us know that you have a question and we'll ask you to unmute and ask it. And then we've saved lots of time at the end to also get to questions. So with that, I'm going to go ahead and introduce today's speaker, Tamsen Peeples. Tamsen has a degree in marine biology from the University of Santa Cruz, and she's a current master's student at the University of Alaska in Anchorage, in Juneau. She has a deep knowledge of commercial kelp farming in the state of Alaska and spent 4 years working for Blue Evolution, helping to design, build, and operate their commercial kelp hatchery in Kodiak. She served on Alaska Agriculture Task Force, and she continues to support farmers and entrepreneurs throughout the state to help grow kelp farming industry here. So it's really a pleasure to have Tamsen with us today. And with that, I'm going to go ahead and pass the floor over to her. Tamsen, I think you may need to unmute.
Yep, just kicked me off the screen for a second. Well, thank you so much for the great introduction. I'm really happy to be here. There is a lot that I'm excited to share with everybody on this whole process. This is really exciting to see more people getting involved with it from the get-go. So is everyone able to see my presentation, the title slide at this point?
We see the back-end speaker view. I think if you can switch the display settings.
Let's see.
On the top there.
Let's see, come on. Why aren't you working? Can we see that now? Is that going?
We're back to the regular. Try if you go to slideshow and then presenter view, sometimes that works.
Let's see. Let me try one thing real fast. I just had to disconnect my secondary screen. Is that working now?
We are still seeing the PowerPoint window.
Ah, interesting. Okay, let me try one more thing. That do it?
Yep.
Okay.
That's great.
All right, thank you for your patience with that. Right, so this is basically, as Lindsay just said, it's going to go over the 3 major species that we've been working with commercially in the state of Alaska and how to collect and transport the material for the commercial hatchery operation. So first I want to go over, for those of you who are unfamiliar with what the life cycle of a kelp looks like. So here's a mock-up of a parent or adult kelp plant. At some point in its life cycle, it becomes a sporophyte, which is the big brown thing that we see in the seawater. Whether that's a big piece of bull kelp or sugar kelp. And at some point during its life cycle, it also produces some fertile material, either a sporophyll on its little side sticking off here to the right, or a sorus on that big blade or big leafy part. And those fertile material releases spores that actually swim around in the water anywhere from 12 to 48 hours, depending on the health of the spores and the age of them. But eventually those little guys run out of juice. They stop, settle, and germinate into tiny little gametophytes, which are only a few cells long, and these are microscopic. Those gametophytes then mature and fertilize one another. So the males fertilize the females, and the females produce a little egg, an oogonium, or an oocyte, that then grows into a teeny tiny little recruit. So it looks like a microscopic kelp blade under the microscope, which then grows into a juvenile plant, which might be a few centimeters long, and then eventually into that big, beautiful, giant, multi-foot long piece of seaweed that we want. So going over morphology, I'm going to be throwing around a bunch of big terms. I'll try and keep them as basic as possible, but I use a couple terms interchangeably. This is a ribbon kelp, an Alaria marginata, one of the species we will be focusing on today. It has a blade, as do most of these kelps, which is, you can think analogous of the leaf. This is where it's doing all of its photosynthesis, turning sunlight into energy. It's usually fairly soft. Sometimes it can be kind of leathery. It's usually brown on these big kelps. And it usually extends for most of the plant. So if you think about the bull kelp, all those long kind of hair-like blades that come off the top of the bulb there. Some species, like the ribbon kelp, have a midrib, which is kind of a leathery section that runs down the length of that blade, and that's to keep the blade from folding in on itself and making sure that it has more surface area exposed to sunlight. All of these kelps will have a stipe, which you can think of like the stem of these seaweeds or these plants. They serve like a stem for a plant that just kind of keeps it up off the ground. And at the bottom of that stipe is a holdfast, which may look like a root system, but it's not serving any sort of nutritional uptake or nutrient uptake role like a root does for a terrestrial plant. These are literally just anchors. These kelps secrete a biosurfactant that's basically a marine epoxy that allows them to stick onto some pretty crazy surfaces and withstand some pretty crazy surf and surge. And then some of these kelps, of the 3, the one that we're talking about, the ribbon kelp, have those sporophylls, which kind of shoot off to the side. Sometimes they can look like a cluster of grapes. And that's where the ribbon kelp produces its fertile material, but the bull kelp and the sugar kelp don't have those, but I wanted to show what those are when I refer to them as sporophylls. So the first type of seaweed I want to cover is bull kelp. I imagine that most people are pretty familiar with what it looks like. It's got that single float or ball called a pneumatocyst, and extending from that is a long cylindrical stipe that extends down to the bottom of the seafloor. And they can grow in anywhere from 10 to 30 feet of water, upwards of over 100 feet of water. That stipe is incredibly long on some plants. They have many of those blades or those leaf-like extensions coming off the top of that float. The blades of these bull kelps can grow up to 6 inches a day. It's one of the fastest growing kelps out there. And on this bottom picture here, the same one I had on the cover, is a blade of that bull kelp with the sori on it. And that looks like the Nutella smears on that blade. And when they're really ripe, they'll actually pull out, those dark sections will pull out from the blade like a sticker almost. Or if you remember as a kid, the fruit roll-ups where you could pull out the pieces of the fruit roll-up. It's kind of like that too. And you'll get multiple Sorus or Sori per blade. So you can see there that there's two in his hands. They develop from the farthest tip down back towards the float. So if you think about the way that kelp grows is kind of like our hair. If the root of our hair is or the head of the bull kelp is our scalp, we grow from the roots out. So the tips are we call the distal ends, or I'll just refer to them as the tips. But they develop the fertility farthest from the float, then back towards the float. And I've seen fertile bull kelp anywhere from June through November before. A lot of these bull kelp plants are annual plants, meaning that they are gone by the time the winter rolls around. But some are perennial plants. You can get the occasional individual that is able to survive the winter, and you can get fertile material on those plants throughout the winter if you know where to look and are able to locate them. The next plant or species I want to cover is that ribbon kelp, Alarium marginata. That's the one that we looked at in the diagram earlier. You can see that mass of sporophylls there at the bottom, and that's zoomed in. The right picture is zoomed in on what the sporophylls look like. These are the ones that have that midrib, that leathery piece that runs down the length of that blade. They have those sporophylls, again, those offshoots that kind of look like grape clusters. They can be long and skinny like this. They can be short and stubby in appearance. There's a lot of variation in that, but they'll always be down closer towards that holdfast or the bottom of the plant. You'll find these in the mid to low intertidal zone. So if you're familiar with going out and tide pooling, they'll be down below the blue mussels. So it usually goes barnacles and that fucus, that rockweed, then the blue mussels, and then below that is where you'll find the alaria or the ribbon kelp growing. These like good current, so often like in channels or areas, it's going to get good water exchange, and they can also handle exposure to waves pretty well. So usually they're on rock walls or larger boulders. And the fertile material is located on those sporophylls, but unlike the bull kelp, they develop from the inner side out. So they're developing from the roots towards the tips. So you can see here that one blue arrow is pointing towards that, again, that Nutella looking smear that's on the edge, the inner edge of that sporophyll. And I've seen these plants fertile from June through October. These again are annual plants, meaning that they will disappear as the fall comes around. But sometimes you'll see just that group of sporophylls attached to the rock with almost no blade or midrib left. And you can get still fertile material and spore release from them through the fall as long as they're still there. Now, Alaria does have a lookalike plant that's called broad-ribbed kelp. It also has a midrib, just like the ribbon kelp. You can see that thick leathery part running down that blade. But unlike ribbon kelp, it does not produce sporophylls. So that's a big difference. If you see one of these plants and it doesn't have those grape-like clusters on the bottom, it's not the right ribbon kelp that you want. This is also found at extremely low intertidal, so way down below the level at which you'll see the ribbon kelp. So if you have a negative, 3 or 4 foot tide, you might see these really down deep or washed up on the beach. Just again, look for those sporophylls because that's where you're going to be collecting your fertile material from. And if there are no sporophylls, it's the wrong plant. So here's a picture of those two plants side by side, just to give you an idea of what they'd look like in comparison. On the left here, we have a ribbon kelp. You can see that thinner midrib and those kind of nice riffles or ruffles to the blade. And on the right is the interloper, the broad-ribbed kelp. You can see it's much flatter, much smoother, and a much thicker midrib. These differences can be subtle, but again, the main one you want to look for is, does it have those sporophylls, that grape cluster at the bottom or not? And then our last species we're going to cover is the sugar kelp, or Saccharina latissima. This has a very finely branched holdfast, that kind of anchor system. It's very delicate in comparison to a lot of the other kelps. And you'll often find it growing on pieces of shell or small rocks on muddy, sandy bottoms. So you can see here, it's kind of intermingled with some eelgrass, and eelgrass also loves that muddy, sandy bottom. The blades are a nice golden brown, and they're kind of crunchy. If you kind of take a piece of that blade or that leaf part and put it, you know, smush it in your hands, it will kind of crunch almost like a piece of paper. And oftentimes it has these bullations that kind of look like puckers running up the length of it. They don't always have the puckers, but it's a pretty common thing to see, those bullations. These are low intertidal to subtidal, so you can find them if you're walking on the beach at negative tides, or if you need to snorkel, they're not super down deep. They're below the alaria or below the ribbon kelp in the intertidal zone. And again, they really like protected waters. They love the sandy, muddy bottoms. They can have some pretty good water flow. So if it's in a channel, it doesn't get any wave exposure, but has lots of water movement, they like that area too. Now the sori, or the fertile material, develop down the length of the blade, starting from that distal end or that tip, like the roots of our hair. And it can be a long stripe like you see in this picture, or it can be kind of big Nutella spread patches. And I've observed and been able to get spore release from these plants from May almost through January. If you can locate a bed of sugar kelp in the subtidal area, as long as you're able to go back there and find some fertile sections, you can get spore release for over half the year. Now, sugar kelp also has an interloper or an evil twin, and that is the split kelp. This is a much thicker, leathery blade. When they're really young, the plants, the sugar kelp and the split kelp, look very similar, but as they become adults, it becomes very apparent which one is which. That blade is much thicker, much leathery, or it often splits. You can see in that second picture where it gets those long cuts down its length. Those aren't made by someone with a knife. That's just how the plant breaks apart naturally in the water. And a big difference to look for is its hold fast. Again, that anchoring system. The sugar kelp has a very delicate, lacy, kind of woven looking hold fast system, whereas the split kelp is much more robust. It's usually shorter and thicker in comparison. And these are deeper than the sugar kelp. And they're often in very exposed areas. So if you see something that you think is sugar kelp, but it's in a pretty wavy area, it's most likely that split kelp. So locating a good bed is kind of your first big step for going into this whole Sorus identification. There's a couple really good resources out there for locating beds. This is probably my favorite one. This is the Mariculture Mapper through the Alaska Ocean Observer System. So I put that web address down there at the bottom. It's in its beta testing stage, but it's free to use. You can log in and save your maps. And if you look here on the right, it has these kelp biobands, and they actually do break them down into some species. Like you can see Alarias marked on some of them. Your dark brown kelps, your dragon kelps, your giant kelps, even your bull kelps are marked down there. So this is a really good resource to just kind of cruise around electronically and see if you can find some beds that have been observed in the past. And this data is coming from the Shore Zone project, which was done in the early 2000s, where they literally flew planes and took video of almost all coastline in the state of Alaska. And so someone had to watch all that video and say, oh, there's bull kelp on this section, and this has been digitized, so it's a lot easier for us to utilize. So I'd recommend playing around with that. You can also adjust your layer types. It's a really, really neat tool to play around with that saves you the time and effort from going out on the water. Another good tool you can use is just your charts, your maps and your charts, because they'll show you certain kelp beds. So if there's a kelp bed marked, that might be a good place to go look for potentially your bull kelp. And then also you can see what substrate, what the bottoms are looking like. If is it sandy and clay, is it muddy? Again, look in those areas that have mud if you're looking for the sugar kelp. So you can look at a lot of charts to really help out before you, or at least identify some potential areas to go check out before you go head out on the water. And what does a healthy kelp bed look like, or what does a healthy kelp bed entail? One of the big things you're going to be looking for to determine if it's a kelp bed you want to utilize a lot is your density. So how many of the plants that you're looking for are there? Is there just one sugar kelp plant? Well, that might not be the best bed to target. If there's just a small handful, maybe it's not the best conditions there or the best stock of material. So look for an area that's got tons and tons of the species that you want. I'm talking hundreds of plants ideally. And then look at the plant condition. Do they look healthy? Are there holes in it? Is it being heavily predated upon by other organisms? But do keep in mind that these plants are, again, annual. So they do naturally degrade throughout the summer. And just like our hair, the tips will degrade faster than the roots will. So just like we get split ends, the ends of these kelps will naturally degrade. And eventually their rate of degradation outpaces the rate in which they're growing. And so they'll just begin to slowly erode away. And then sometimes you'll see epiphytes, different organisms, and sometimes different seaweeds growing on the kelp. But the good news is that epiphytes aren't really a big deal for us when it comes to collecting seed stock. So on the left here, we have an image of bull kelp with some other seaweed growing on it. And that won't really affect our seed stock because that's not going to be coming into the hatchery with us. As long as we can get clean, fertile material off those blades, it doesn't really matter what the parent plant looks like. And these two images on the right are bryozoans, which are encrusting animals that will just grow directly onto the kelp. Again, it's not a big deal if that parent plant has bryozoans on it. Even if they're on the sori directly, they can be very easily removed at the hatchery with just a straight razor. It's not a big issue. Again, if that's all that you have available to you, it's not a big problem for us to clean that off when it gets to the hatchery. And selecting the best parental stock, the nice thing is we can be choosy and you can really look for what you want on the farm. So we can be selective in choosing what we want the parent plants to look like. So if we want big bladed alaria, so we want that nice broad leaf section to that alaria, you're going to be looking for parent stocks or parent plants that have that big leafy section on it, and in hopes that the offspring will look like that as well. Though I do want to note we're still studying that association of parental phenotype or what the parents look like and what effect that has on the offspring. I've personally seen some variability from season to season, and we're not sure if that's because of conditions or just one parent plant was more effective or at survival than the other ones, and it just looked a certain way. But it is nice that we can be selective, especially if you find a big bed with, hundreds, if not thousands and thousands of these parent plants. You can really sit there and pick and choose which ones you want your offspring to look like. And how much to take from the bed. The nice thing is we don't have to take the entire plant with us. You can just cut out that fertile section, whether it's the sporophyll or the sori. This allows the rest of the plant to continue to grow and reproduce. So you're not removing that entire organism from the genetic pool, which is really important to appease fish and game and also just to maintain that wild kelp bed. This also means it's a very low impact. Again, we're not removing dozens and dozens of organisms. We're just taking a small section of that plant away. And it's a heck of a lot easier to ship just pieces of bull kelp than 50 full bull kelp plants. Though that being said, I do recommend that you send along 5 to 10 of the full adult plants. So, 5 or 10 full pieces of saccharina or full pieces of that ribbon kelp. Maybe not the full bull kelp, because I think we all know what bull kelp looks like, but maybe a section of that blade along with the Sorus, just so we can make sure that we have, number one, the right species, and then we can make herbarium samples from those. So just plant pressings, and that allows us to do genetic research on them at later dates. And so how do you collect it? There's a little bit of different ways to do it depending on the species. So the ribbon kelp, you just pop off that sporophyll, or that little grape section that looks the most fertile. So again, that has the darkest Nutella looking section on it. For sugar kelp, you can just cut out that dark section with a knife or a pair of scissors. For bull kelp, it's very similar. You're just going to be pulling off that blade and then either pulling out that fertile section if it's really, really ripe, or just snipping that section out from the rest of the blade with a pair of scissors or a Vicky knife. While you're doing this collection, you don't have to be instantaneously packaging it and making it look really pretty right away. Just throw them into a bucket and then you can do a mass processing later. As long as it's being done within the next couple of hours, it's fine. And then just make sure that you know which beach you're collecting this material from. So if you're going to be collecting, say, bull kelp from multiple different bays, keep those separated. But you don't have to log the exact GPS from where you took them. Just make note of like, oh, this was collected in Windy Cove, or if you have a GPS app on your phone, it's pretty easy just to drop a pin on that. And then you can use that information later on your permit, which I'll cover in a little bit. One of the challenges that we face, or there are a number of challenges that you'll be facing through this, is locating the beds, especially sugar kelp, because it can be kind of tricky to find those nice, muddy, quiet, calm areas where the sugar kelp likes to grow. And then making sure that you know when the plants are going to be fertile. As I mentioned earlier, there's a pretty substantial range in fertility dates. You know, maybe that's May through October or May through January. Really, the only way you can really keep an eye on that is to go out, identify your kelp beds, and just keep an eye on it visually throughout the summer. Maybe every month or so, go out there and take a look at those plants to see if they're getting fertile. And not all the plants will be fertile at once either. There will be some staggering, just like not all blueberries become ripe at once. And Fish and Game requires us to collect at least 50 parent plants per species from within 50 kilometers of where that farm is going to be. So at times, locating 50 fertile plants can be difficult. Like I said, they're not all going to go fertile at once. There's kind of a big bell curve, and you're just going to have to, through observation or luck, just manage to time out so you know where and when to be to collect those 50 fertile parent plants from within that 50-kilometer range by water. And I also get asked, what's the easiest way to do this? Do you want to be doing it in wetsuit snorkeling, or do you want waders to be walking around in them? And honestly, my preferred method is really dependent upon which species I'm going for and what my tides are doing. You know, it's kind of a pain in the butt to get in a wetsuit or a pair of waders and to go stumble around on the rocks. So if I'm going for a high intertidal species like that ribbon kelp, and I've got like a -2 foot tide, I can just go down there in my rubber boots. But it's my preferred method, honestly, if I don't know what the tides are doing or if I have to get wet, is just a wetsuit with a pair of Grendons over the top, just to protect the wetsuit from the rocks. So you can just kind of wear, whether they're fishing wading boots or tennis shoes or whatever you have on your feet, and then just to be able to walk around on those rocks without getting hurt. And if you know, I've gone out in waders before, and you're looking at a perfect plant that's right at your feet, but if you bend over and stick your arm down, it's still too deep. So you're trying to have to like sit there and kick up the plant with your foot to try and reach it. And I just realized that it's a lot easier just to go in something that can completely get wet than just your, you know, lower body. The permits that you're going to need for the collection will be this seed stock acquisition and transportation permit through the Alaska Department of Fish and Game, aquatic stock and acquisition and transport permit, pardon me. There used to be 2 different permits. Looks like this year or last year is the first year they've combined them into one, which is very nice. You can see here that you're just going to be putting down the name of the collector and the applicant. Those can be different. So the applicant could be the hatchery operator, and then a collector could be whoever's going out on the boat. I would recommend that you get this application in at least a month prior to when you need to be collecting. They can do pretty fast turnarounds, but just to make their life easier, I try to get in about a month beforehand. There's not really an expiration date on them. So if you think, oh, I'll probably be going out to collect sometime around mid-August or late August, it's beginning of August, I'll put in my application now, and then it'll be good for the rest of the calendar year. And then you will need to be listing everyone associated with collection. So if you're the one collecting and your name's not on that permit, that's going to be an issue. So just make sure that if you're the one going out there and putting hands on the kelp and doing the harvest, that your name is on the application. And you can have multiple collectors per application. So what do you need to go out and collect? Number one for me is just your trusty bucket. That's kind of the biggest tool that I always use, is just make sure you've got a bucket. And then a knife or a pair of scissors. That could be a Vicky knife, or if you're more prone to potentially stabbing yourself like me, just a pair of household scissors works great. For doing the processing of the kelp, which I'll go over shortly, you'll need paper towels. It's nice to sometimes have rags or towels you don't mind getting covered in kelp, slime, and seawater, just to kind of keep the kelp moist while it's being worked with and transported around. And then having a Sharpie to write the information onto the Ziploc bags. And then I'm not sure if the group here has hobo loggers, but it might be something to consider. And I might be able to loan a couple from the lab that I work in now, but they're temperature recorders. So they're these little pendant loggers that are about the size of your thumb that you can just throw into the cooler or the bag with the kelp, and it just records the temperature for upwards of hundreds of days. You can adjust how frequently they're taking the temperature. So that's one way, just to make sure that you're keeping the kelp at a constant temperature or a relatively constant temperature throughout the entire process. So let's make a kelp burrito. This is kind of what the kelp would look like when you're done processing it if you're shipping whole plants. So the first step is to just lay down paper towels. These are just your normal Bounty, whatever brand from Costco or their local grocery store you want to get. It doesn't have to be anything fancy. Lay those down on a flat surface, and then you're going to lightly dampen them with seawater. So that can just be as much as flicking it a little bit with your hands, or if you're really worried about not getting it wet enough, dunk that paper towel in seawater and then really wring it out. You don't want that seawater or that paper towel to be dripping wet. That's going to be too much water for the kelp. So you're going to lay that damp piece of paper towel down on the table or the top of your fish tote or whatever it is. And then you're going to lay out the piece of seaweed, the Sorus, the sporophylls, whatever it is, on top of that paper towel. And you can have multiple pieces on that paper towel as long as they're not touching or overlapping. This is just to prevent them from basically becoming anoxic or not having good airflow between them, and they'll kind of get icky and degrade faster if they're overlapped or touching. So once you have that paper towel surface covered with your kelp nicely separated, you're going to put another layer of paper towels down, dampen that with some seawater, put more seaweed down, put more paper towels down until you've got a nice big chunky almost sandwich, and then you're just going to roll that thing up and stick it in a plastic bag, again labeled with what species it is and what beach or cove you collected it from. And then shipment is pretty easy. Once it's nicely packaged in that Ziploc bag with its paper towels lightly dampened with seawater, they are pretty happy for a long time. I've had kelp shipped all over the state back and forth it's been sitting in its little plastic baggies for upwards of 3 or 4 days and did great. And I was still able to get spore release from it when I was running the hatcheries. Some tips for shipping it though: you want your insulated fish box or a cooler, whatever you have available. If you're willing to part with the cooler for a couple days or weeks, that's great. It's reusable. If not, an insulated fish tote works just great. You want to place those cold packs, those frozen cold packs on the bottom, and then insulate those with a layer of either bubble wrap or just some pieces of cardboard so that those bags of kelp won't be touching directly onto that cold pack. We want to make sure that that kelp inside is not going to freeze. And so then you stack up all of your bags in there. If you've got room at the top, you can place some more bubble wrap or some cardboard and put some cold packs on the top as well. And then seal it up. Slap that with a whole bunch of do not freeze stickers, but keep refrigerated stickers, and then gold streak it. I've FedExed them before. I've taken them as checked luggage before. Again, they can travel for a couple of days. I know that Dr. Steckel was concerned about X-ray exposure to these plants, but I've had perfectly fine spore release and offspring from X-rayed plants. Gold streak does not X-ray the plants. And unless you're going to be flying it with it, it's a really good option for flying Alaska Airlines from one location to another. Again, days at a time, it's fine in these coolers. But if you're really worried about storing that material for a long time before you have to ship it, so say you went out and you collected all your material, it's in your bucket, and now you come back to your home port to start packaging it, and oh no, a huge storm's come in, planes aren't flying, or you can't run it to the hatchery. What are you going to do with this stuff that you just spent a whole day collecting? If you can keep them in chilled seawater, especially circulating chilled seawater, so even if it's a bucket overflowing continuously, in theory, that stuff will hold for a long, long time. I've held parent plants in tanks with just flow through chilled water in the lab for weeks or months at a time, and they do fine. So whether that's, constant flow through of water, or if you can have a bucket with seawater just sitting outside with a towel over the top of it just to keep the seaweed underwater, and then maybe every 12 hours or something change out the seawater, that works well too. But again, if they're in that little paper towel system already and you think that you're going to be able to fly within the next 24 hours or 48 hours, it might be worth just to process them that way and just keep them in stasis. Again, I've kept them in that way in the Ziploc bags and paper towels for days at a time and still had good spore release at the hatchery end. Rule of thumb, if your seaweed's turning green, it's not healthy and is either dead or dying. That could be from either being too wet for too long or too dry for too long, especially if it's in those Ziploc bags with no water exchange or air exchange. So if you're really worried and you want to keep checking on your seaweed, you're welcome to keep cracking open those Ziploc bags and taking a look at them and seeing if they're turning green. But it's kind of a fine line with how much moisture you have in those bags. Again, we don't want that paper towel in the Ziploc bags dripping with seawater. We want it just damp enough that they're not going to dry out or desiccate, but they're also not going to start decomposing in there. Additional resources for identification. If you're out in the field and you get stumped onto which plant you're looking at or forget which rules of thumbs to look for, there's Seaweeds of Alaska and seaweedsofalaska.com. So one's a printed field book and one's just an online resource you can access through your phone. That's probably the most complete catalog of Alaskan specific seaweeds, or what we have here in the state. Another really great app that Lindsay turned me on to recently is the Seaweed Sorter app, which is really cool. You have to pay for it. It's not a free app. I think it's like $4. But it really allows you to key in. So it'll ask you questions like, is your kelp brown? And you say yes, no, or I don't know. And it helps you identify down to a couple potential species that you're looking at. So it gets kind of nitty gritty, those details of like, what does the holdfast look like? Is it robust or is it delicate? And that can help you determine down between some of those interloper species. And I know I've just thrown a whole bunch of information at you, but I'm happy to answer any questions, to go back and review any of the material we just covered, go in more in depth. I am at your disposal. So thank you everyone so much.
Awesome. Thanks, Tamsen. That was super helpful and really informative. Yeah, do you want to go ahead and come off of screen share and then we can see everyone in gallery view and maybe have a bit of a discussion?
Sure.
Awesome. Great. Yeah. So everyone feel free to switch to gallery view and mirror view so you can see everyone and ask Tamsen any questions that you might have come up. There wasn't. Matthew, go ahead and unmute. I see your hand, Matthew, but I think you have to unmute yourself in the bottom left where there's the mute key.
That was by accident, actually.
Oh, okay. No worries.
I'm pushing buttons here.
Sure. Does anyone else have any
take that off?
Questions? I think you, okay, got it. Yeah, great.
I have a couple.
Sure, go ahead.
First, about the AOOS mapping software that was done in the early 2000s, as you said, this is more a broader question about how kelp beds operate, but how much should we expect to see kelp beds change or move over time, especially with some of like the warming water issues that we're seeing elsewhere? Yeah.
Great question. From my experience, just what I've seen in the past 6-7 years, going back to the same kelp beds over and over, I haven't seen a whole lot of variability. You might see some variability in how long the kelp's hanging around in the fall. Like on really hot summers, I've seen the ribbon kelp kind of disintegrate a little bit faster, for example. But you go back again the next spring and it's still there. Thankfully, we haven't had the huge uptick in warming temperatures like they have in Southern California with their kelp beds just disappearing. We haven't gotten to that stage yet. More what we're seeing is some of the warmer weather species, like we're seeing an increase in the macrocystis, that giant kelp spreading up through Alaska. But the kelp beds that are already here seem to be sticking around pretty good so far.
Another question I had was about handling, like should we be wearing gloves when we handle the Sorus? How much can it really take in terms of active handling? Like once we collect it, should we just like try not to touch it at all? Any of those things, do we need to sanitize the bucket ahead of time? If we're going to put it in seawater, do we need to make sure that seawater is filtered? Those type of things, like how much do we have to be worried about contamination factors?
Sure. In the field, the nice thing is number one, we're working with mainly intertidal plants. So the sugar kelp and the ribbon kelp are both used to being exposed to air throughout their life cycle and throughout the seasons. You think about how much they can take on a single day where it's 80 degrees outside. They can go from being in 40-degree water to 80-degree air and still survive. So they can take a bit of abuse when it comes to handling them, which is great for us. They're not the most delicate plant to work with. In terms of sterility and maintaining that factor, that's more on the hatchery side. Once it gets to the hatchery, that's where they're going to be wearing gloves. They're going to be using the filtered seawater because they want to try and prohibit or remove anything else from growing in the hatchery but that one seaweed. There is a potential to, if you pull a piece of kelp and it's covered in bryozoans, if you have the time while you're in the field, you can just scrape those off with like the back of a Vicky knife even. But again, that's something they can also do in the hatchery side. The main thing you really want to make sure is that when it's being shipped, that it's as happy as it can be. So again, maintaining that moisture level and that temperature level for that kelp. So it's going to have its best chance at getting to the hatchery in one piece and also reproductive and able to spore out. So you don't really need to worry about keeping it sterile. You don't want to be putting it into a bucket that's been holding oily rags or anything, but it doesn't need to be, you don't have to wash it out or bleach it or anything beforehand.
Great. Thanks, Tamsen. Looks like Grafton has a question.
Yeah, hi, thanks, Tamsen. I had a couple questions about, you were mentioning that some of these species, the three that we discussed, can survive for more than a full cycle. Is that just bull kelp, or can you see ribbon or sugar survive the full season?
Great question. I've never seen ribbon kelp survive a full season, but I have seen sugar kelp down deep. It'll the blade will degrade down until it's basically just the stipe and then like maybe an inch of the blade, and it gets really thick and leathery and dark. And again, that's stuff that never gets exposed to the intertidal zone. And then I've seen that grow back again next year. But for the most part, rule of thumb is that these plants will disappear in the fall. I'm sure if you spent any time out on the water in the winter, you've seen the really, really big bull kelp bulbs and still surviving out there. But most of those kelp beds just kind of disappear with the winter storms.
And with the ones that do survive, have you seen any difference in the sori? Would they be more or less viable to go for like a second year plant? Or
it's a great question, and I'm not sure about that. I've talked with a kelp operation down in British Columbia that seems to prefer the second year plants just as a final product. So they say that it's going to be a better kombu, but in terms of its viability for sporing, I have never really considered it or heard of it, but it's a great question.
Okay, thank you.
Mm-hmm.
What other questions do folks have? There's lots of material that came through today.
I have one more. I'm wondering with ribbon kelp and bull kelp, if we're just taking a single sporophyll and a single blade off of that, and that's enough, or do you recommend us taking multiple of each?
I I would, you know, if you have the option and if you find a really nice ribbon kelp plant that has like 5 or 6 really promising looking or really dark colored sporophylls, just grab them all. As long as you're making sure that you have material from at least 50 plants to satisfy that fish and game requirement. The hatchery will always, what will happen when they get to the hatchery is that they'll clean those blades off as much as possible. So they'll, you know, they might be scraping them with a straight razor just to remove any epiphytes on there. They might be doing a betadine rinse to try and not quite sanitize, but kill off anything else that's growing on top of them. And then they'll dry them out and spore release all together. So all of the plants, all of the Sorus material from each species will be released together and be mixed all together. So if you can get multiple sporophylls from a single parent, that's great. It's not going to do any harm. And it might be a better boon just to have extra material. On the hatchery side, it's way better to have too much spore release than not have enough material.
Do we need to be concerned about like taking too much from a single kelp bed and like potentially impacting that bed, that wild kelp bed in the?
Again, that's a nice thing about just taking the fertile material away. Most of these plants will have multiple sori or multiple sporophyll throughout its growing season or throughout its fertility season. So if you're taking 2 or 5, even out of 40 sporophylls from a single plant, those sporophylls will eventually become fertile as well. For sugar kelp, if you're taking just 1 or 2 of those big patches, those big Nutella spread patches off that plant, that rest of the plant from the roots up or the holdfast up will continue to grow and then will continue to become fertile later on. So as long as you're not, you know, clear cutting basically a whole kelp bed and just removing the fertile parts that you need, and then again maybe those 5 or 10 ID species pressing plants, that's very, very low impact.
Hey, Tamsen. Hey, this is Jeff. How are you?
Good. How's it going, Jeff?
Good. Crazy morning. Sorry I didn't get to listen in again. But we're talking about doing some direct seeding. It was counseled that we should delay processing the plants as long as possible. How long can we store these plants prior to induce them to release? Can we just store the
if you're
kelp?
collecting? Yeah, it's pretty easy to, if you're collecting the full parent plant, for example, if you're collecting 50 full parent plants, you can pretty much store those in the flow-through tanks for a while. I've had, due to season timings and just storms or what other factors for on the research side of things, so not commercial, I've been able to go out and collect, been like, oh shoot, here's 5 or 10 adult plants that aren't quite fertile yet, but I want to grab them before they get tore up by a storm or before this next big storm comes in and I'm not going to be able to come back out. You can bring those parent plants back into the lab, store them in a flow through tank, like if you have a living stream, just with flow through seawater, you can actually monitor its fertility development that way. But I've not yet really found a way to push pause on the development of the fertile material. So if your Sorus is already there, it's going to release its spores no matter what you do, from my experience. So if it's already fertile, you kind of just got to go with its natural fertility. But if you go out there and you find, especially for research scale stuff, if you don't need those 50 parent plants, if you find 5 or 10 plants that are becoming fertile but aren't quite there yet, you can definitely bring that stuff back to the lab and hold it until it becomes fertile.
And then I guess since I have the floor, what if we want to go to a large scale tank? It doesn't seem like having the material to, we dilute it and throw away a whole bunch of it, right? We're not really concerned about the number of spores we actually have. Isn't that right? When we go, if we want to, we're thinking about doing some PVC tubes that are either 3 or 4 inches in diameter in a bigger tank and go flow through. So, you know, it would just take everything that we're doing with the 2 inch and, you know, double the diameter, and hopefully the string length is another 6-fold or 8-fold in length. We should have plenty of spores that do that.
You should
right?
have, yeah, especially depending on how good your spore release is and how many parent plants you're bringing in. On the hatchery side, as long as you're maintaining that sporing density, so where you count your spores, you figure out how much water your pipes are going to be in while they're being inoculated, and then you can calculate that sporing density. And if you have a good release, you might only need milliliters worth of spore solution for 40, 50 liters of water. It really just depends on how good of a spore release you can get. And there was a question I saw in the chat about the full plant samples be shipped in separate cooler with similar treatment. Those full parent plants can be wrapped up in that same kelp burrito or in the same Ziploc bag, or if there's not room, a separate Ziploc bag, and just be shipped the same way. So again, you just want to make sure that they're not being stacked together directly on top of each other, that they've got that insulation layer, that separate layer of paper towels between them. And then they can just be rolled again as a burrito, stuffed in a plastic bag and shipped in the cooler as well. Great question on that one.
Tamsen, have you had any experience with there being a better result if you pull parent material from multiple kelp beds versus collecting your 50 specimens if you find a really large kelp bed, like you were describing with 1,000 plants?
So far, when I was doing all the commercial stuff, it was more of a collection for convenience and utilitarian. I haven't done a lot of experimentation with collecting from different beds or seeing different fertility rates across different regions. But from my experience, the one that seems to matter the most is just finding plants that are fertile at the same time, and then have the shape and size or the leafiness of what you want. So finding those parent plants that you say, that's the stuff I want to be growing on my farm, and being able to collect that one. And I've seen the most variation in what the plants look like in that ribbon kelp. Sugar kelp and bulk kelp, it seems to just grow the same year after year, but that ribbon kelp, I've myself witnessed quite a lot of variability in how it looks from season to season, both the farms and the wild beds.
Thanks.
Any final questions?
Awesome. Well, thank you, Tamsen, so much for sharing your expertise with us. It's so great to have you on the call and be able to pick your brain. There were a couple of questions in the chat about accessing the recording. I will share the recording with the Native Conservancy and CRRC, and they can share it out with their networks, which I'm assuming is how most of you got the invitation. But if you want to make sure you get a copy, please send me a direct message in the chat, and I will make sure that your email's on the list, and I can give you a copy of the recording. I want to make sure everybody has this guidance in hand when they go out again later this summer and into the fall. Awesome. All right. Well, with that, I will hang on just to make sure I receive emails from everyone. But thanks, Tamsen, for being here, and thanks everyone for joining.
Thank you so much for having me. It's been a pleasure.